Hi, I am Aaron J.D. Pabilo.
And I am Justin Escobar, and we will discuss
the cell cycle and malfunction during cell cycle. First, what is cell cycle?
The cell cycle is a carefully orchestrated series of events that culminates in a cell division. It is broadly divided into two main phases, the interphase and the mitotic phase.
there are two phases of cell cycle, the interphase and the mitotic.
Interphase, often called the resting phase, is anything but inactive. It's the longest part of the cycle, a period of intense preparation for cell division. During this phase, the cell performs its normal functions, grows, and crucially duplicates its DNA.
Interphase is further subdivided into three distinct phases.
G1 phase. This is the cell primary growth phase. It synthesizes protein, produces energy, and carries out its specialized function inside the nucleus. DNA exists are relaxed chromatin. Each chromosome consists of a single chromatid. A crucial checkpoint, the G1 checkpoint, monitors the cell size, DNA integrity, and the presence of necessary growth factors. If conditions aren't right, the cell can enter a resting phase called G0 or undergo apoptosis or a programmed cell death. S phase. This is where DNA replication occurs. Each chromosome is duplicated, creating two identical sister chromatids connected at the centromere. The amount of DNA doubles but the number of chromosomes remains the same. This precise replication ensures that each daughter cell receives a complete and identical copy of the genetic material. The G2 phase. The cell continues to grow and synthesize proteins necessary for cell division, such as those needed for spindle formation. Organelles are duplicated and the cell prepares for mitosis. Another critical checkpoint, the G2 checkpoint, ensures that DNA replication is complete and that there is no DNA damage.
Next is the mitotic. It consists of two main processes, karyokinesis and cytokinesis.
But first, what is mitosis? It is a process of cell duplication or reproduction during which one cell gives rise to two genetic identical daughter cells. These are the four stages of mitosis.
First is the prophase.
Chromatin condenses into visible chromosome in the prophase. The nuclear envelope break down and the mitotic spindle begins to form from the centrosomes, which might mitigate the opposite pole of the cell.
Next is the metaphase.
Metaphase. Chromosomes align in the metaphase plate and an imaginary line in the middle of the cell. Spindle fibers attached to the centromeres of each chromosome. The metaphase checkpoint ensures proper chromosome alignment before proceeding to the next stage.
Next is the anaphase.
Sister chromatids are pulled to opposite poles of the cell by shortening spindle fiber. Each chromatid is now considered an individual chromosome. Telophase. The nuclear envelope reforms around each set of chromosomes at the poles. Chromosomes begin to decondense back into chromatin.
Finally, cytokinesis occurs, dividing the cytoplasm and creating two distinct daughter cells. In animal cell, this happens through the formation of a cleavage furrow, while in plant cell, a cell plate forms. Cell cycle controls and checkpoints. The cell cycle isn't a free-for-all. It's tightly regulated by a complex network of proteins, including cyclins and cyclin-dependent kinesis. These proteins control the progression through the checkpoints, ensuring that each step is completed correctly before moving to the next. Spindle Assembly Checkpoint. Spindle Assembly Checkpoint, or the SAC. This checkpoint occurs during metaphase and ensures that all chromosomes are completely attached to the spindle fibers before anaphase begins. This prevents unequal distribution of chromosomes to daughter cells. DNA Damage Checkpoint. This checkpoint monitors DNA integrity throughout the cell cycle. If DNA damage is detected, the cell cycle is halted, allowing time for repair. If the damage is irreparable, the cell may undergo apoptosis or senescence, a state of permanent cell cycle arrest.
Proto-oncogenes.
These genes normally promote cell growth and division. Mutations can turn them into oncogenes, which are permanently switched on, leading to uncontrolled proliferation. The example is RAS and MYs.
Tumor suppressor genes.
These genes normally inhibit cell growth and division or promote apoptosis. Mutation that inactive tumor suppressor genes remove these breaks, allowing uncontrolled cell growth. The example is P53, RB, BRCA1/2.
DNA repair genes.
These genes are responsible for repairing damaged DNA. When these genes are mutated, DNA damage accumulates, increasing the risk of further mutations in proto-oncogenes and tumor suppressor genes. The example is MSH2 and the MLH1.
Telomeres and telomerase.
Telomeres are protective caps at the end of chromosomes that shorten with each cell division. In most normal cells, this shortening eventually triggers cell cycle arrest. Cancer cells often reactive telomerase, an enzyme that maintains telomere length, allowing them to divide indefinitely or immortality.
Angiogenesis.
Cancer cells stimulate the growth of new blood vessels, or the angiogenesis, to supply themselves with nutrients and oxygen, further promoting their growth and spread, or the metastasis.
The loss of contact inhibition.
Normal cells stop dividing when they come into contact with each other. Cancer cells lose this contact inhibition, allowing them to grow in disorganized masses, or the tumors.
Evading apoptosis.
Cancer cells often develop mechanism to evade apoptosis, allowing them to survive even when they are damaged or should be eliminated.
Metastasis.
The ability of cancer cells to spread to distant sites in the body, forming secondary tumors. This is a complex process involving multiple steps, including detachment from the primary tumors, invasion of surrounding tissues, entry in blood or lymphatic vessels, survival in circulations, and colonizations of distant organs.
Genomic instability.
Cancer cells often exhibit genetic instability, characterized by an increased rate of mutations, chromosomal abnormalities, and changes in DNA methylations. This instability contributes to the rapid evolution and heterogeneity of tumors.
Inflammation.
Chronic inflammation can promote cancer development by creating a microenvironment that supports cell proliferation, angiogenesis, and metastasis. Cancer is not a single disease, but a group of diseases characterized by uncontrolled cell division. Normally, cells grow and divide in a regulated cycle. In cancer, this cycle is disrupted, leading to continuous and abnormal cell proliferation, forming a mass called a tumor. Now, let's turn our attention to chromosomal abnormalities. These occur when there are changes in the number of structure of chromosomes, the structures within our cells that carry our genetic information. Humans typically have 23 pairs of chromosomes, totaling 46. These abnormalities often arise from errors during cell division or exposure to teratogens, substances that can cause birth defects. Karyotyping process of photographing and arranging chromosomes helps identifying these abnormalities. The most common disorder of trisomy is Down syndrome, wherein the 21st chromosome has 3 instead of 2 chromosomes. Most cases of Down syndrome are not due to inheritance but random mistakes during formation of reproductive cells of the parents. Turner syndrome, a condition that affects only female as a result of one of the X chromosomes is missing or partially missing. The physical manifestation of Turner syndrome is web neck, short structure, swollen hands and feet. Some have skeletal abnormalities, kidney problems, or congenital heart defect. The physical manifestation of Klinefelter syndrome, its primary features are infertility and small poorly functioning testicles. Sometimes includes weaker muscle, greater height, poor coordination, less body hair, breast growth, and less interest in sex. Trisomy X syndrome, characterized by the presence of extra X chromosomes in each cell of a female. Patau syndrome, caused by having an additional copy of chromosome 13 in some or all of the body cell. The physical manifestation of Patau syndrome is clenched hands, cleft lip or palate, extra fingers or toes, hernias, kidney, wrist, or skull problems, low set ears, small head. Edwards syndrome, caused by having additional copy of chromosome 18. The physical manifestations of Edwards syndrome is cleft palate, clenched fist, defect of lungs, kidney, and stomach, deformed feet, heart defects, low set ears, severe development delays, chest deformity, slowed growth, small heads, and small jaw. Both cancer and chromosomal abnormalities highlight the delicate balance of our cellular and genetic processes. Understanding these conditions is vital for developing effective treatments and providing supports. Understanding the cell cycle and its control mechanism is crucial for understanding cancer development. By studying these processes, scientists are developing new strategies for cancer prevention, diagnosis, and treatment. Thank you for joining us on the journey into the fascinating world of the cell cycle. Stay curious and keep exploring the wonders of biology.